Well, hey biology honors, hope you're doing well when you watch this video. I am going to do my best to walk you through the last topic and the last concepts for the test on Friday. Just a disclaimer, my voice is still a little under the weather, so I'll do my best for this. But definitely watch this if you're out for track or if you just need a refresh on what we cover on block day, okay? So first off, we're going to start with genes and variation. We have just a few things to discuss before we dive into the last couple of concepts. So first off, Darwin, when he developed his theory of evolution, he didn't understand how heredity worked. And so this left him unable to explain sources of variation, like where does variation come from, and how do those traits pass from one generation to the next. Of course, that's Mendel's stuff. And so Mendel's work, dove into that, opened up, scientific eyes to how traits are passed on. But genetic terms, evolution is any change in frequency. So that's what I want you to start thinking about, okay? Any change in frequencies is evolution, okay, in alleles. And so natural selection acts directly on phenotypes, not genotypes. So, for example, animals can't see the genes, of course, when they eat another animal. They're just looking at the phenotype, not the genotype. And so vocabulary, so a population is a group of individuals of the same species living in the same area. We've talked about this a long time ago. But populations change, not species, right? So populations evolve, not species. So a couple of terms you need to know about. Gene pool right now is the sum of all copies of all alleles at all locations. Loci means locations in a population. So here's just an example of some warthogs, and you have all these different alleles. That is the gene pool. And so if you have a different mouse population, when scientists try to determine if a population is evolving or not, they look at allele frequencies. And so that's exactly kind of what they look at, brown versus black alleles, okay? And so alleles, again, if you don't know or remember, alleles are one form of a gene. And so how many times does that happen? So genetic variation, yeah, mutations can happen that stopped, production of pigment. There's that white deer, methicillin resistant, Staphylococcus aureus, MRSA. Puggle is a result of breeding of a pug and a beagle. My roommate actually had a puggle in college, him and his girlfriend. So sources of variation, first off is mutations. Of course, mutations can have variation, any change in a DNA sequence. Genetic recombination, this is a result of sexual reproduction. That's also a source of variation. And then lateral gene transfer as well. This happens in prokaryotes where bacteria transfer DNA. Think about our transformation experiment with Griffith, right? Bacteria can pick up DNA from other bacteria, here's that conjugation tube, and they can exchange DNA with each other. That also leads to variation. So some traits are single gene traits, like controlled by one single gene with two alleles, like widow's peak, hitchhiker's thumb, tongue rolling. And some are a lot more complicated than that. Here's a single gene trait. But some are polygenic, which means they're covered, controlled by many different genes. And so you can see like the curve here. It's usually a bell curve, right? So if you think about this is a single gene trait, and this is a polygenic trait, okay? So here's where the last little topic gets into, okay? We need to be able to explain natural selection, how it affects single gene and polygenic traits, which I already kind of talked about, but we'll look at it a little bit more. Describe genetic drift and explain how different factors affect genetic equilibrium. So for example, natural selection on single gene traits can lead to changes in allele frequencies and thus changes to phenotypic frequencies. So here's the initial population. The other ones died. Maybe they were super easy to see. Maybe for whatever reason they died. But as you can see, after 30 generations, the majority of the lizards are darker color. And then in class I'll ask, why are some of these reasons? But for those who are watching the video, maybe they absorb more light and therefore they have more energy. Maybe they can blend in better. Who knows? But there's an advantage there. And so this is evolution, changing in allele frequencies. And so natural selection acts on phenotypes, not genotypes. So again, if a predator is going for this mouse, it doesn't see if it's homozygous dominant or heterozygous. It just sees a mouse. So natural selection acts in three quantitative, which means numerical ways. And you need to know these for the test. Stabilizing, directional, or disruptive. Stabilizing favors averages. So let me see if I can write here. Average. Directional favors one direction, so either in one direction from the average. And then disruptive is away from, directions from the mean. So away from the mean. I'll show you what this means here in a second. So here's some graphs to show you that. So this is stabilizing. We want the average. Directional means it shifts one way or another. Think about beaks, right? If I can try to write with a mouse, think about beaks and finches on the Galapagos Islands for directional. And then you have disruptive, which means it goes against the average. And I'll show you an example of this in a second. So first off, stabilizing birth weight. This is purifying selection, so we want an average birth weight. What is this? From a textbook that's roughly 8 years old, roughly around 7 pounds is an optimal birth weight. 7.5, 7 pounds. You get too large, the body has problems. You get too small, the body has problems. So you can see that there's a higher percent of mortality, sadly, when someone's born prematurely or when someone's born really, really heavy. And I forgot the world record for birth weight. It's something like, I don't know, something like 14, 15, 18, it's something crazy. You can look that up if you desire. In directional selection, individuals at one extreme have higher fitness. So for example, if this plant is being eaten by this warthog, a number of spines, like sticky, I don't know if you ever stuck yourself on a rose, but spines can be a survival advantage for plants. The more spines, the less likely it is to be eaten. And so if it shifts this way to more spines, that is directional selection. This is also an example of directional selection. Longhorn cattle, they're longer horns. Longer horns offer protection for the new calves. And so baby calves, and so those longer horns allow for protection, but the longer the horn is, the better the protection. So that's directional. And then finally, disruptive is opposite extremes. We don't want the middle, we want opposite extremes. So an example of this from another textbook I have is bill sizes and the black-bellied seed cracker. So for example, small birds can feed more efficiently on soft seeds. Large-billed birds can crack hard seeds, but where they live, there's not much in the middle. So the birds that have like the medium size lower bill, like they can't really do either one well, and so they have a lower fitness level. Okay, genetic drift is another topic that we need to talk about. And genetic drift is random changes in allele frequencies from one generation to the next. So this is just random changes. Again, according to evolution, everything's random. That's not faith-based according to the Bible. According to the Bible, everything, God is sovereign. He's in control. But nonetheless, death by chance. So these butterflies die by chance. Okay, so all these alleles, all these genes are lost. So you have all these butterflies here flying around. And so there's two different examples of this. First off is bottleneck. So an environmental event results in the survival of just a few individuals. So for example, if I were to ask you, can you describe this, each red bean is alleles and each yellow bean is alleles, well, you can say like, man, it's pretty even, right? 50-50. But afterwards, the surviving individuals, just due to chance, they're mostly red. And so see how that affected the population? Mostly red alleles now. And so that's a population bottleneck. Of course, biblically speaking, Noah's flood, population bottleneck. Greater prairie chickens was one of the most popular birds in North America, roughly 100 million in 1900. But there was fewer than 50 in the 90s. That's a population bottleneck, okay? Another example of this is elephant seals. They're hunted to near extinction. Population increased to 20 individuals in the 1800s, give or take. And so those 20 repopulated. So today's population, 30,000. But here's the problem. When you get that low, you lose genetic variation. You lose the ability to adapt when the environment adapts. Okay, and so again, population bottleneck, you have all sorts of equal percentages here. Look, they're all 20%, all these alleles. But afterwards, something random happens and you completely lost the blue allele. It's no longer in the new population. And so that is an example of how evolution can change just due to random chance, allele frequencies. The next one is founder effect. Like if you're founding a colony. And so this is genetic drift when changes in allele frequencies, when a few individuals colonize a new area. I don't know if you've ever met an Amish family. They're super nice. But sometimes they have a higher rates of certain disorders than the general population. Why? Because there's a lot more genetic variety. So you found a population, you don't have much genetic variety, and so look how different the two allelic frequencies are between both populations. This actually has an amazing example, one of my favorites in science. This is Tristan da Cunha, if I said that right. And so basically 15 British colonists founded this settlement. But here's the thing. One of the early colonists apparently carried a recessive allele for retinitis pigmentosa, progressive form of blindness. And so in the 1860s, 4 people had this really rare disease. Why is that? Because this is the founder effect. The frequency of this allele was much higher in that population. And so it's just an example of how this can actually work in real life. You can kind of read through this about other examples, founder effects, maple syrup urine disease, Mormon communities, Amish communities. This is all examples of that. Okay, so how do new species arise is the last thing we're going to talk about. So where do new species come from? Okay, should be a from there. But so what type of isolation? So there's a couple of different ways new species can come onto the scene. Again, God created according to kinds, not species. So the formation, technically, let's define it first. Speciation is the formation of new biological species. Remember, species are individuals that can reproduce and produce fertile or viable offspring, which means the offspring can produce more offspring. Horse and a donkey can make a mule, but mules usually can't make more mules, so therefore horse and donkey, not the same species. Biblical worldview, God created according to kinds. Young Earth says roughly 6,000-7,000 years ago. Old Earth would say something different. But biggest thing about Genesis, who is God and who are you? You're created in the image of God, super important. And so as creationists, we must frequently remind detractors that we do not deny that species vary, change, or even appear over time. Species and created kind are not synonymous. So 3 different ways this happens: geographic, behavioral, and temporal isolations. When you isolate species, if you give them enough time, they can form subspecies and perhaps even speciation. Okay, so 3 different ways this happens. Behavioral, which means mating rituals are different. Mechanical, which means structural differences prevents mating. And then prevention of gamete fusion. Gametes of one species function poorly, so this could be different numbers. And then hybrid inviability, which means hybrids can't produce viable offspring. So like the donkey and the horse makes a mule, that's hybrid inviability, okay? So reproductive isolation, post-zygotic isolating mechanisms operate after fertilization. So this is the hybrid, right? So mating happens, fertilization happens, but yet the resulting hybrid remains infertile. Sometimes this can be a lower fitness level. Maybe it's a miscarriage. Sometimes the hybrids don't survive very well. And this is a post-zygotic. A zygote is a fertilized egg cell. Post-zygotic isolation. So back to our isolations. Behavioral, mating. It's all about somehow, some way, they differ in some sort of activity, and therefore they no longer mate together. They no longer interbreed. Geographic. Famous example here is 2 squirrels when the Grand Canyon separated. They live on different sides of the canyon. Geographic is when something is physically separated. And there's a picture there, north rim versus south rim. And there's just another example. And then temporal is time. So one form of cicada emerges every 17 years. The other form emerges every 13 years. So they are not even on the scene at the same time. So therefore they have reproduced at different times. So therefore they have temporal isolation and developed into different species. Example of this is also the frog. Okay. And so just 2 further terms. Allopatric speciation occurs when populations are divided by a physical barrier. So just know the definition there. Physical barrier is the key word there. And then, so for example, those 2 squirrels, the canyon became too deep, so now you have 2 different squirrels there. And then sympatric speciation occurs without a physical barrier. This is kind of rare in as far as animals go, but very common in plants. So for example, polyploidy. We did our strawberry lab. Remember that lab? And strawberries have 8 copies of a chromosome set. 8 copies. They're 8 in. They're octoploids, which is crazy to think about. So polyploid happens often in plants. They can use that to their advantage, make different genes, make more of a gene, etc., so on and so forth. But this can actually cause speciation. So a tetraploid plant might arise if chromosome separation and errors occur in meiosis, right, non-disjunction. And so therefore, the next generation, this tetraploid plant can no longer mate, of course, pollen, seeds for production through animals. Diploid plant they no longer can mate together. So if they produce a sex cell, it's only 1 copy, but this one has 2 copies now. And so therefore, there's a non-viable or infertile triploid plant.